Thanksgiving, what a great time to talk about this next group of macromolecules, and those are proteins. So we're going to talk about some protein foods and why we need protein in our diets. Proteins are the most abundant of all the biomolecules that we've looked at. They are probably the number one in terms of responsibility of all the biomolecules. What do they do for us? What do they do in other living organisms? They have a really diverse range of things that they do, including regulating body processes. Remember homeostasis, this idea of regulation of so many things such as our heat and cold mechanisms, things that regulate our temperature, our diet, everything. Structural functions, building different structures such as our muscles, which are protein. Protective functions, immune system, transport, enzymes, and toxins. So we're going to talk about these. Enzymes are so important. You're going to hear us talk about enzymes a lot this year, but the reason they are is because they are proteins and they are the workhorse of the body. They are able to act as catalysts to speed up a lot of different chemical reactions, which can include reactions that break things down. And that term there you see, catabolic, the breaking down of substrates. So an example of that would be taking some polysaccharide and breaking it down into individual monosaccharides. It's involved with anabolic processes, taking the raw materials, the different monomer units perhaps, and building something more complex. And the term catalytic is what it is really referring to, how certain enzymes, that is their job, is this ability to catalyze reactions. And so they show you here an example of this enzyme that is involved with the catalysis of maltose to glucose products. Here's some examples of some of the enzymes and their function. So we see digestive, transport, structural, etc. We'll go into some of these in some detail, but for now I just kind of take a look at this list. See a lot of different functions, a lot of different examples of different proteins. And you recognize at the top, these ones we've discussed, these are the enzymatic proteins that involve with digestion. And there are others, but these are some of the big ones. And some of you might have cued in on this one. Insulin is involved with regulation of the blood sugar in our body, and we'll see some others. So all proteins are made up of these monomers, and the monomers are amino acids. And amino acids have this basic setup, and this you want to know. So it'll have a central carbon. This is where the arrow is, and this, they call it the alpha carbon. They will have it'll have a hydrogen attached to it, and then it has this thing called an R group. An R group is a whole bunch of other things, and we're going to look at those, but it's a side chain. And that R group is really what establishes what is this amino acid. And then it has our friend, the carboxyl group over here on the right. Carbon, double bonded oxygen, and then an OH here, so this whole thing together, carboxyl. Remember that gives it the acid component, so amino acid. And then over here is an amino group, hence there's the amino part, with nitrogen and 2 hydrogens. There are 20 of these that are common in most of the proteins that we are familiar with. And each one of the amino acids is going to have a different R group, and we'll go to those. But it's the R group that tells us a lot about what the chemical nature is of that particular amino acid. And these are going to be some of the things that R groups are going to confer on these amino acids. So some of them will be nonpolar and aliphatic. Some of them will be polar. Some will be positive, negative, and some will be nonpolar aromatic. Now you remember aliphatic were the chains when we looked at hydrocarbons before, and aromatic were the rings, in case you couldn't remember that. Here they are. I think we need to create a song for this to remember some of these amino acids. But here they're grouped according to the nature of their R groups. So where you see the blue boxes is really showing what the R groups are. So the simplest one up here on the very left is glycine. That's an amino acid. Notice that it's drawn in a way that you can see that the side chain is on the bottom. That's just hydrogen for this one. And then the amino group up here, notice it has a plus charge on it. And look over here at the carboxylic acid group has a negative charge. And the good news is that's true everywhere you're looking here, pretty much. Maybe with, no, actually, I'm not even sure there's an exception. Because what they're showing you is in the body, that COOH, which would have been the carboxyl group, because this is the acidic part, is going to drop a hydrogen. And guess who picks it up? The amino group. So the amino group gets a positive charge on it from that positively charged hydrogen, which is a proton. And then the carboxylic acid group has a negative charge on it because it is dropped off the hydrogen, meaning it's doing what acids do, and that's putting hydrogen ions into the environment. So all the ones up on the top left are just, as you can see, just chains of mostly carbons and hydrogens. Occasionally there's a sulfur that's in this one here. So these are the aliphatic ones, because they're mostly hydrocarbon chains of some length or another, not particularly long. Then you go down below and you see the polar uncharged R groups. Up here we see the positively charged R groups. So you can see the plus charges here on the part that's in the blue box. Remember, those are the R groups. Negative charged ones. And then at the very bottom, the nonpolar or aromatic R groups. Now, you'll recognize some of these, and some of them you've never heard of before, but we'll talk about a few of them as we go here. So amino acids have their own little code names. So, and this is what this is all about. So valine, you could also just use this code name, just V, or you could use the abbreviation VAL to represent it. Interestingly, they don't all totally make sense, because aspartic acid, its little code symbol is just the D or ASP. Now, if you ever, anytime we'd have to decode something that had a bunch of amino acids, I'd have to give you a key for that, so you don't have to worry about memorizing all those. But it's just so you can recognize that there are these ways of showing symbols for these particular amino acids. There are a few, though, that are called essential. And what we mean by an essential amino acid is it's something that we have to get in our diets. That means that we have to get foods that contain them. Where our body can make other ones without that, they don't necessarily need to get it in our diets. And so we're going to look at these here very shortly. Now, just like every other thing we've talked about, when we have the monomer units, the single units, amino acid, one on the left and two on the right, and they're going to join together to form something we're going to call a peptide bond. So the name, just like we were talking about saccharides for sugars, here we have a term called peptides, which is relating to proteins. So a peptide bond right here is the union of these two individual units of amino acids through the same process we talked about before, dehydration synthesis. So polypeptide is lots of them together. And most proteins are pretty big. There's a lot of polypeptides. You could have a dipeptide, meaning only two, but a lot of them are quite large. Proteins are huge, actually, relative to a lot of other molecules. And we're going to talk about how they're made later on when we look at this idea of what's called translation. Don't worry about that one yet. Now, they throw in a word here. They say that proteins are often combined with non-peptide prosthetic groups. So if you think of a prosthesis as something that would be like if somebody loses their hand, they might get a prosthetic hand. So it's kind of like an addition onto the original. There are groups of atoms that would be called prosthetic groups that sometimes can join onto these protein or polypeptides. There's a nice little video here I encourage you to go to. This is from OpenStack, so you should be able to go here and see protein synthesis. Well, that's a topic on its own that we'll get to later. But this we're going to do in class as well as just get a little preview of it here. And so proteins start as being just a long chain of amino acids that get put together by peptide bonds. But then something kind of wild happens, magical, and it's that these proteins, these long strands, start to fold and become different over time till they ultimately reach the shape that they're meant to be. Remember, shape is destiny? Well, that's very true for proteins. Here's the primary protein structure for this particular insulin. It says it's a bovine serum insulin, so this is something you would find in cattle, and it's a type of long chain of amino acids. And this one has an A chain, and it has a B chain, and then those chains are linked, and you can see where they're linked here by these things right here. So the first structure of a protein, the primary structure, is just the big long chains. Okay, just a series of amino acids linked by peptide bonds. We'll look at this in a little bit, so we can, it's going to determine what that protein is that primary structure. So the order in which all those amino acids come together and bond will determine the actual end product, the actual protein at the end, once everything has done its thing or folded. They give us an example here that I'll get into later in class of sickle cell anemia and what can happen if just one little mistake happens within that long chain of amino acids. This is showing what sickle cells look like. Again, I'll talk about these more in class, but what you can see is there's some abnormal shaped red blood cells here which can't act like the normal ones. They can't carry the oxygen that they need to. Secondary structure, you're going to get some folding, and that folding is going to be either called an alpha helix or a beta pleated sheet. We're seeing a lot of alphas and betas lately. Helix is just what you see up here. It's just like a spiral staircase. And the beta pleated sheet is going to be more like a zigzag kind of a thing, like a pleat, like in your skirt. I'll show you how this is done when we get to class, but mostly what this is involved with is hydrogen bonding. So after you get this long strand of amino acids or polypeptide, then you start finding these things folding or spiraling as hydrogen bonds form between parts of these long chains. Tertiary structure is where we look at those R groups and we see what they're doing, how they interact. And we'll see these more specifically also coming up. Here's an example. So not only do you have the long primary structure, the chain of amino acids, but then you also have the hydrogen bonding, so that's secondary, and then certain things are going to do some other connecting between parts of that long, now folding molecule. And then finally we get to quaternary structure, where several of these kind of polypeptide groups can get together and form a much bigger unit. So here are 4 levels. Primary, string of amino acids with their peptide bonds. Secondary, forming these pleated sheets, these beta pleated sheets, or the alpha helices, these helixes. Tertiary, you get more interaction within this, but it's these R groups that we mentioned that are going to interact. And then finally, quaternary, groups of those tertiary structures together. And that's it for now.